Alright, now I'm going to talk about DNA cloning procedure. So now we want to multiply the DNA by the help of the bacteria. When the bacteria multiply, it will help us to multiply our gene of interest as long as we transfer our genes into the bacteria. Now this is called as DNA cloning. And if we're going to transfer into bacteria, we will need the help of vector. There are many vectors. You can either use plasmid, liposome, or virus. But the most common vector that usually the researcher use is plasmid, the most common one, and also the most easier one to help to transfer into bacteria. So you can't direct transfer the genes into bacteria. You will need the help of the vector that help you to transfer it into bacteria cells, and then the bacteria will help you to transcribe and translate the DNA into protein. So the first step is always extract the plasmid. So you will need to get plasmid. Now you can also, don't want to go for extractions, you can synthesize artificially in lab, synthesize the plasmid. But at a lower cost, this one extractions will have lower cost compared to synthesized artificially plasmid, uh, or the buy from the biotechnology company. Now, extractions of plasmid is very easy. You got the bacteria cells, you culture them, make them keep on replicates and multiply to high amount so that they will have more and more plasmid. So they will have many cells, more plasmid. Then you extract this plasmid out and you will get the plasmid extractions and purified in a microcentrifuge tube. So you have these tubes here that contain plasmid. Now how you're going to do the extractions? You must need to use something that remove their cell wall and remove the bacteria cell membrane, and that is using the lysozyme and some chemical. So after you remove it, if let's say this is a test tube, you put in the lysozyme inside and put it all the bacteria inside here. After you incubate with the enzyme, the membrane here already removed by the lysozyme, removed by the chemical like SDS, you will have this, you will need to separate them and purify them. So how is by centrifugation. So you spin it down, spin it this whole tube in a centrifuge machines, and you will get them fragmentized into two different level. So at the bottom here is stores that are heavier, and at the above supernatant you will contain a lot of plasmid. So you transfer this plasmid into here, into a new tube, transfer the supernatant into a new tube. So you got the plasmid purified without all your cell membrane, because all the cell membrane is going to sink at the bottom. It is more heavier, and all the organelles will be at the bottom here. The plasmid will be at the supernatant, and you just take the pipette out the supernatant into this tube. So you got the purified plasmid inside here. But now you want this plasmid to carry your genes. But if the plasmid is in a circular structures, you can't have a gene being transferred in. So the next step is this test tube. You want to open out the plasmid. How is again by the help of enzyme. And this enzyme is called as restrictions endonuclease, or RE, and restrictions enzyme. So now let's not draw so much of plasmid inside. We draw a representative plasmid. This circular or double stranded plasmid is going to open out, and they might, because you use RE, it is going to produce a sticky end. Now the plasmid already opened out, is ready to take in the genes. So where you can get the genes, you also need to extract the genes. Now again, this one, you can go for traditional method to extract the genes or to extract the DNA that you want to clone to transfer into the bacteria. Or you can synthesize the genes. Synthesize meaning that you just do it artificially in lab using some chemicals. Or you can go for reverse transcriptase technique. If you don't want extractions, you can just go for extract the mRNA and then convert back into cDNA. But we will go to the common method where you just directly extract the genes out. Now this one, extractions of genes, you can take it from the animal cells. Let's say you want the human genes to be transferred, so you must get the human cells DNA extractions. If you want firefly genes to be transferred or the tiger genes to transfer, so you just take their cells out and then extract the DNA. Now this DNA is inside this tube. After you have DNA extractions, you will have the whole DNA inside here. So this is extractions. Now we don't want the whole DNA. We only, out of DNA is very long. Out of this long DNA, you might only want one small segment of the DNA sequences, maybe these regions. So this is your gene of interest. You only want these regions. How you're going to get it? Is again, you will need scissor to cut it. And that scissor is called as RE. So you're going to put the RE into these solutions. Once you put the RE inside, then you will get your DNA that is restricted. So this is the microcentrifuge tube, and now you got your genes out. Again, sometimes it depends on what type of restrictions enzyme that you use. Some of the restrictions enzyme might produce sticky end. So let's say in this case, you have a sticky end, you use the sticky end restrictions enzyme. So now your genes will be like this. Now in this case, this second step is always restrictions enzyme digestions. And you must use the same restrictions enzyme because they will cut and produce the same complementary end so that they can complement to each other when you mix them together. So it is important to write in same restrictions enzyme. Now the next step is you want to put now you can put your genes into this plasmid. So how? By mixing these two tube solutions together. And ligate. So the third step is ligations. So you put these two solutions into one test tube that contain the plasmid here, and this is the genes that is being ligated inside. Now you will need the help of enzyme. Again, you must mention the enzyme name, that is ligase enzyme. You can talk about this is the DNA ligase enzyme that will add into these solutions and enable the ligations of the plasmid and genes of interest. Now you will have another thing. This one we don't call it as plasmid anymore. We call it as recombinant plasmid. When you talk about this recombinant plasmid, you know this is the plasmid that carry the genes of interest. The next step is you want to transfer the recombinant plasmid into the bacteria. So the vector will help you to put and transform inside. So the next step is always transformations. Transformations is a very easy and fast process. So once you already got your recombinant DNA, the next step is, let's say this is your microcentrifuge tube again. This is your recombinant DNA. You must mix them with bacteria. So this is bacteria. Now how you can mix them? You just mix them like this. most probably you get nothing. The plasmid will be still in solutions. The bacteria will not have any recombinant plasmid in. You will need to use one method is called as heat shock method. Heat shock means that you are just shocking these bacteria with some heat, with higher temperature for a while, for a few seconds, and then remove them from the heat. Then they will take in the bacteria. Now this one, the heat is not so high, it is only 42 degrees Celsius, and they must be needed to appear or occur in a calcium chloride solutions. So once the bacteria that you have cold it or freeze it, you quickly put them inside this test tube, mix with the recombinant plasmid, and then put maybe in a water bath that has 42 degrees Celsius. And inside these solutions, you might contain calcium chloride, so you can add some calcium chloride inside. And after this heat shock, maybe for 30 seconds, not even until 1 minute, you can remove it, and the heat shock is actually help to open the pores on the bacterial surface so that it can allow the plasmid or recombinant plasmid to went inside. So when it become porous at these 30 seconds, they will take out the plasmid, and you will got these results. So you will see this one. The bacteria that contain the recombinant DNA. Now, if 100% of bacteria take it, then you don't need to do the next step. But in fact, in the reality, whenever you do this heat shock technique, you only have 1% of your bacterial populations inside these solutions that take out the plasmid. Not 100%, only 1%. So imagine inside these solutions, you have thousands of plasmid, you have thousands of bacteria. You might end up only have 10 bacteria that taken out the plasmid. So you have two possibilities. One is the bacteria that is totally have no plasmid at all, and another one is the bacteria that contain your gene or interest. So this is the one that we want. This is the bacteria that we want, but how we going to differentiate them? So this one will be followed by clonal selections. You want to select which bacterial clone that is contain the gene of interest. But how to select is all depends on the plasmid that you have used. So inside a plasmid, they not only have the origin or replications, promoter, restriction site with your gene or interest that can be incorporated inside. They will have these sections of one segment of the DNA that contain marker genes. Now, marker genes will play a role in clonal selections. There are many different types of marker genes here. Let's say in this case, the marker genes used is ampicillin resistant genes. So meaning that during the clonal selections, you will need to throw the whole thing of your transformations tube, the whole solutions into petri dish. Now inside the petri dish, you have agar that is provide nutrients for the bacteria to grow. So you're going to spread the solutions up here and then let it incubate for overnight. Now in this case, let's say your plasmid here contain your marker genes. The researcher use is the one that contain ampicillin resistant genes, meaning that those bacteria that contain plasmid, they are ampicillin resistant. They contain ampicillin resistant genes. So in this case, the agar block must be added with ampicillin antibiotic. Now this is usually they can use the nutrient agar that provide the growth of the bacteria. They must add with ampicillin, and those bacteria they grow up inside here. So when you spread it, you might only have one of the bacteria here, and then after you incubate overnight, you know the bacteria every 20 minutes they will start to divide. So you will have more, and then keep on have binary fissions, and you have many clone of them keep on multiply and finally form a clonal. So we call this as clonal selections. Now here also, when you spread, you have one bacteria, and one bacteria. And if you let it incubate overnight, they will start to multiply. They are exactly the same as the parent cells. If the parent cell contain ampicillin resistant genes, the whole bunch of these, whole clone of these will contain the ampicillin resistant genes. They will contain your genes of interest also. So they are just the asexual reproductions, produce the genetically identical offspring. The longer that you put, the bigger is the clone here. Now, so this enable you to select which clone that has. So those that contain ampicillin resistant genes, they are going to survive in ampicillin antibiotic plate. This one cannot survive because they don't have ampicillin resistant genes. So this one, they will die off. So there are no clone of them added here. And after this, after clonal selections, you already eliminate all those that do not have the ampicillin resistant genes. You can just pick up one of these and go for mass productions. So mass productions, you can take one clone of these bacteria and you inoculate into a fermenter that you provide them a lot of nutrients, more oxygens, and then maybe you put with aerations, and you can extract the protein out. So you will have all these bacteria will then happily multiply inside with the genes of interest being transcribed and translated, and they will start to produce protein out into these solutions. So the next thing, once your protein is inside here, let's say your genes that you clone is human insulin genes. Now you have a human insulin protein that is produced in these solutions. You are going to take out these, purify these solutions. So you remove all your bacterial cells and purify it, the insulin hormone, and then you can inject into patients, into diabetic patients. Okay, so this is the overall view of clonal DNA cloning. Make sure that you know that DNA cloning into bacteria, you will require plasmid as vector. Later, we will look into how the bacteria can be used to transfer the genes and how the liposome can be used to transfer the genes. Now, however, if you know your target is bacteria, if you wish to transfer the genes into the bacteria, then you must usually use the plasmid. So you must get the plasmid out from the bacteria, extract the plasmid, and then you cut them with using the same restrictions enzyme to those enzymes that cut your gene of interest. And then they can produce the complementary sticky end. So if this one is AAA, then this one you will get TTT. So they are complement to each other, and you need to paste them together to form recombinant plasmid by the help of ligase enzyme. So you just put the ligase enzyme, they will automatically help you to seal the phosphodiester bond between the plasmid and the gene of interest. Now for extractions of plasmid, how you're going to extract is by putting some enzyme like lysozyme to break the bacterial cell wall and put some chemical inside that going to remove all the cell membrane and organelles out, going to separate and release the plasmid into the solutions. And you need to centrifuge, meaning to spin down so that you can get the purified plasmid, and all the cell membrane organelles will be stay at the bottom because it's more heavier. So you transfer the supernatant into this one whole new tube that is a purified plasmid. Okay, once you got the recombinant plasmid, you want to transform them into bacteria. It can be done easily using heat shock method, meaning you apply a heat for maybe 30 seconds in a solutions that containing calcium chloride. And now it will open the pores of the bacteria. The recombinant plasmid can be go inside. And after heat shock for 30 seconds, remove it, the pulse of the bacteria will resolve. So your plasmid, recombinant plasmid will stay inside the bacteria. However, heat shock technique didn't guarantee 100% of the bacteria will take out the plasmid. Only 1% of the bacteria inside these solutions will take out the plasmid. So always need to know that inside one solutions you have thousands of bacteria, thousands of plasmid. Now you want to differentiate which bacteria that have the plasmid, which one have not. You need to depends on gene marker that the plasmid use. If the plasmid contain gene marker that is comprised of ampicillin resistant genes, then you throw them into ampicillin antibiotic neutron alga. If they might have multiple gene marker here. If they have another gene marker that is tetracycline resistant genes, that is the genes that confer them to be able to survive in tetracycline antibiotic. Resistant means resist to antibiotic. You know antibiotic will going to kill the bacteria if they don't have if they do not have resistant genes. So in this case, if they have some more the tetracycline resistant genes, then you need to put in the tetracycline antibiotic here. Only those that contain the plasmid, they can survive. So this helps you to select the appropriate clone that contain the recombinant plasmid that has also means that it will contain your gene of interest. Then only you go for mass productions. Select this confirmed clone for replications. If you didn't do this test, if you didn't do the clonal selections, you would multiply both of these bacteria. Some that will contain recombinant plasmid and some of them that do not contain will be get multiplied if you didn't go for this step. If you directly go from this step into mass productions. So for mass productions, you provide them more nutrients, provide them more oxygens, more aerations, and then you can have bacteria multiply to thousands to many of them, and then you will have them to help you to transcribe and translate the protein. So the protein can be expressed and can be released out into the solutions. Then the next step is you will do the purifications of the protein. Remove all these bacterial debris, bacterial cells, and get the pure protein out. So this is a general procedure for DNA cloning.